get started, everyone. Thanks again for being here for another week of the seminar series. So I'll just have my little introduction here, and then we'll get into Anna's research. So my name is Robert Driver. I'm a postdoc at Duke University, and welcome to the AOS Student Research Grant Seminar Series. And before we get to our speaker today, I just want to introduce the series and the grant that is the foundation of the series. So each year, the AOS offers funding to students and early career researchers with the AOS Student Research Grant, and the AOS Student Research Grant Seminar Series celebrates the challenging and innovative work of the AOS Student Research Grant recipients by sharing the funded work to AOS members. And each week we showcase the research or research plans of a 2024, 2023, or 2022 AOS Student Research Grant winner in a seminar style format. And we'll have one seminar each week for 15 weeks until December 15th. Actually, I just have to make a note here. Next week, we will not have our seminar series. The seminar is moved until December 15th. So we're going to go an extra week at the end. We had to move next week's seminar. So just a quick announcement there. I want to thank everyone for being here and to thank the AOS and AOS Student Affairs Committee for support and input, and especially thank Anna, our speaker today. So before we turn our attention to Anna's research, I want to mention if you have questions, there will be time for a Q&A session at the end of the talk. You can participate either by raising your hand and I can unmute you if you would like to speak your question, or by typing anytime during the hour in the Q&A box or in the chat box. So those three ways. And I'd like to remind everyone this is an AOS sponsored event and that all participants are required to follow the AOS virtual meeting event Code of Professional Conduct. And I'll post a link to that in the chat, as well as a link to the AOS Student Affairs email if you would like to contact AOS Student Affairs, and a link to the post-seminar evaluation form where you can fill that out and help us improve the seminar series with your feedback. Okay, so for today, Anna Rose is currently a master's student at The Ohio State University in the School of Environment and Natural Resources. And I just wanted to note that it's exceptional that Anna is a master's student because she is the first master's student, I believe, in the seminar series to present her master's research, and is currently a master's student. So Anna's academic career began with a Bachelor's of Science in Forestry, Fisheries, and Wildlife at The Ohio State University. And here, Anna researched habitat selection in overwintering white-throated sparrows in the lab of Dr. Christopher Tonra. And in 2023, Anna began her master's degree researching parasite loads, particularly ticks, in a variety of species, including wood thrush, ovenbird, worm-eating warbler, and red-eyed vireo, who we can see on this slide here, many of them, maybe all of them, in the lab of Dr. Stephen Matthews. And in addition to the AOS Student Research Grant, Anna also received honorable mention for the NSF GRFP. And Anna has already presented multiple times at the AOS conferences, including in 2023 and 2024, as well as the 2024 AFO, Association of Field Ornithologists meeting. So with that, I will turn it over to Anna for her talk entitled Tick Infestation Consequences for Ohio Appalachian Breeding Birds.
Thank you so much, Robert, for that lovely introduction. And as we saw at the end there's a lot of tongue twisters in this presentation. So if I have to repeat my words, well, you might hear them repeated. But I am so excited to present for you today. And I'm going to start out not directly with my research, but a little bit about my story as a bird scientist, as an artist, and also as a young person simply interested in nature and that always wants to be outside. So this is my presentation on how I started as a nestling, and now here I am working on my master's as a bird professional. And I'd like to thank AOS for having me today. I'm so excited to be here. Starting out, when I became a nestling, I banded my very first eastern bluebird at the age of 8, and I was immediately hooked. I raised poultry in my backyard with my parents' permission, and from then on, I wanted to band birds and I wanted to be active in the field of avian ecology. I was lucky enough in my undergraduate at Ohio State University to be involved with the Dr. Christopher Tonra Lab of Avian Wildlife Ecology, where I worked on a project looking at overwintering habitat selection in white-throated sparrows. So this was a project I did during the winter compared to my master's, which takes place during the spring and summer months here in Ohio. And this was a wonderful opportunity because I actually got to present this research at my very first American Ornithological Society meeting in London, Ontario in 2023. And not only did I make great connections in the scientific world, but I also got to present myself as an artist and make connections there. And it was actually a very serendipitous meeting that I made connections with Miguel, the Latin American coordinator, for Environment for the Americas. And after seeing my portfolio, it was because of the connections I made at AOS that I got invited to be the World Migratory Bird Day artist last year. And this simply jump-started me into not only being dedicated to my own scientific work, but also in being an artist as well, including having my very first show this past summer here in Columbus, Ohio. Now you might be thinking, Anna, you're an art nerd, you're a bird nerd. Where did the ticks come into this? And I'm so glad that you didn't ask because I'm going to tell you a lot about ticks today. But it was a special opportunity in 2022 when I got to work with someone from a different university, Dr. James Marshall, and he suggested that we work on a project looking at the Lyme disease causing pathogen Borrelia burgdorferi on birds on the Lake Erie Islands. And if you didn't know, Ohio does in fact have islands off the coast of Lake Erie in a very interesting island chain. So I got to stay up there for the summer, capture birds, hone my mist netting and banding skills, and also do a lot of lab work in order to test for this pathogen. And it was during this time while I was doing this research and doing my literature search and learning about the human impacts of tick-borne diseases and also how birds are involved in that cycle, then I started feeling a little not great. And something happened. I got Lyme disease. And this made the project not only something of great interest from a scientific perspective, but also quite personal. And it's something to this day that I still think a lot about, and I have to think about my own limitations in the field when it comes to long-term impacts. But it was during this time, learning about tick-borne diseases, learning about their connections with birds, and how it was impacting my own health, that I realized just how passionate I was about disease and parasite ecology. So when I had the opportunity to do my master's again here at Ohio State, I knew I wanted to do something that was not only pulling my creative and scientific interests together, but also something that I had personal investment with. And I got to take my hodgepodge of ideas and present them to my wonderful advisor, Dr. Stephen Matthews, who helped hone my interest into something that would become the project that I present to you today. Tick infestation consequences on one of my favorite suite of species in the whole world, Ohio Appalachian breeding birds. And I also had a kick-ass committee, Risa Pesipane, a lauded tick ecologist, the amazing Dr. Christopher Tonra, an avian ecologist I worked with in undergrad, and Matthew Schumar, president of AFO and one of the best field biologists I've had the pleasure of working with. And now, getting into our project, we start with why are we studying birds in an avian ecology project? Well, first of all, when it comes to the human disease side of things, tick-borne diseases are on the rise. And not only in the US and here in my home state of Ohio, but also globally. It's interesting to note that this is not a disease that is simply endemic here to North America, but in different ticks of the same genus that we'll be looking at in this project today, they all carry the same bacteria causing Lyme borreliosis that we call Lyme disease based on its symptoms and the pathogen that is caused by. So different ticks actually can carry the same pathogen, which is interesting to note. But in our project, we're just going to be looking at ticks that are not necessarily native, but are present here in Ohio. And Lyme disease itself, which is more of my focus, there's been a 48-fold increase in disease cases in the last 15 years. Back in 2010, we only had 37 cases in the whole state of Ohio. Compare that to last year, where we had over 1,700 cases. And in 2025, we have already surpassed that number as of mid-August, and we're not even through the peak season for human disease cases. So you can see here that we're having a lot of human disease cases here on the rise. And though some of this is caused by better detection of Lyme disease and our doctors and professionals knowing that this disease is becoming more prevalent in Ohio, but it's also simply because we have more ticks on the landscape, and those ticks have a higher prevalence of the bacteria in their system that could infect us. And now you might be wondering, okay, if it's not just detection, why are these zoonoses becoming a bigger issue today? Not just Lyme disease, but a lot of different tick-borne diseases. Well, there are some factors with our landscape. As our landscape changes and as our climate changes, this impacts populations of ticks and wildlife hosts. In particular, adult ticks are overwintering more frequently because of higher low winter temps, which means that the adults aren't dying off as much and can survive with the disease in their system to reproduce another year. This also has something to do with the different densities of mammals that we have in our landscape. In particular, in suburban areas, we have lots of white-footed mice, which are highly proficient at not only carrying the bacteria, but spreading it to ticks that then have it in their system that can bite us. And there are also a lot of factors. This is a topic we could talk about for the whole time that is incredibly interesting, but those are just a few little hints of knowledge to tell you why tick-borne diseases might be on the rise today. And now, because it is of personal importance to me, and I think it's as important as scientists and also as the general public to keep each other safe, I want to talk to you a little bit about tick safety. If you look at the figure at left, you can see the circle without humans and our pets. Zoonotic cycles simply exist on their own without us. So Lyme disease has been present in the environment. I think it's been documented in human specimens over 5,000 years ago that were found in the ice. And the spirochete itself, the bacteria, existed for 60,000 years. But now that we're also on the landscape, ticks can bite us, and we can become a part of that cycle as well. So we have to know how to protect ourselves. Now, this information here is a bit more specific to the Northeast, particular for deer ticks, but this can apply to most other tick species. I like to be aware of my environment, what I'm wearing, and then aftercare for myself after I go into the field. For your environment, it's always good to watch where you are. Stay on a trail unless you have to be me and go out and do a bunch of fun bird work. Be aware of your clothing, how you're wearing it, and if you're able to treat your clothing and prepare before you go into the field. And there are also a variety of EPA regulated repellents that you can use as well. My only recommendation on that is that if you, like any medication that you would read for directions of how to use, it's important to read pesticide labels that not only keep you safe, but also the environment. And of course, always remind people to tick check. And sometimes those pesky ticks will sometimes get through our defenses. So it's always important to know just kind of what it might feel like to have the early stages of some of these tick-borne diseases, which to me felt more like fatigue and joint aches without upper respiratory symptoms. So if you're in an area like the Northeast or the Pacific Northwest, it's important to be mindful of these. However, I found that in the last 2 years of doing field work with putting precautions in place with my technicians and we've been safe and tick-free the last 2 seasons, which has been a wonderful breath of fresh air. But our vials are full of ticks for our study, which is exactly what we want. And now, back to our Ohio tick ecology. Ohio is lucky enough to have more than 12 tick species, and we're finding more all the time. Ohio is only 5 medically relevant tick species for us, but there are actually 7 others that feed only on wildlife and are present in our environment and sometimes on our birds. Ticks all have, or not all, I should say, but the ticks I'm working with have three life stages, with the youngest being larvae, and then they hatch into nymphs and then adults. And only in this last adult stage do they differentiate into males and females. For our project, it's important to note that ticks must blood feed at every stage, which could be our birds at the larval and nymph stage, in order to molt into this next life stage and eventually become adults. And those adult females will also have to take a blood meal in order to then lay their eggs for the next generation of ticks. Now, pulling our bird hosts into it. Birds not only carry and transport these diseases, or I should say, not only carry our ticks, but can act as vehicles for tick range expansion and disease transmission. So, what birds have that's unique that our mammals and other hosts don't is that they can fly long distances. So, oftentimes, our neotropical migrants are responsible for bringing ticks that then drop off into novel ecosystems. So, a lot of the research and literature search that I have done has been on tick presence on birds during the migratory season, simply due to the fact that there is a bit more relevance to the human disease side of things as ticks are being transported to new areas. However, ticks can be present on the landscape all year round, and I, in particular, will be looking at ticks during the breeding season on birds. And one of the questions I often get are, what's the big deal? Birds and ticks have coexisted for over 100 million years. In fact, there have been records of ticks found on warm-blooded theropod dinosaurs back in the Jurassic period, which is pretty awesome. So the relationship between birds and ticks is as old, if not older, than birds themselves. Well, in the last few decades and few hundred years, we've had remarkable changes to our climate, which has resulted in novel ticks here in Ohio that may have never been present here historically, as well as ticks in denser numbers that also was not the case historically. And of course, our birds are also undergoing some new types of stressors that they might not have historically, excuse me, experienced as well here on our field sites. And of course, there are factors that we might not know. So even though this is an extremely old evolutionary relationship, times are changing, and as the environment changes and our densities of ticks and what species of ticks are present in the environment, so could the physiological impacts on birds change. And for our breeding birds, many of you already know that a lot of our breeding species are under duress, with our eastern forest birds experiencing a 17% reduction in biomass. And if you look at ticks adding to this equation, although these effects on adult birds are rare, ticks can cause infection, stress, and even lethality. In addition to our adults, nestlings and juveniles can also be negatively impacted. The reason, or one of the reasons I think that this has not been studied in our eastern forest birds as much is typically tick numbers on these birds are low. Where physiological measurements are looked at on birds is typically in colonial nesting birds, where the birds are dense, close together, and it is much easier for ticks to feed and higher numbers of ticks. So physiological effects on birds have been documented during the breeding season in some of these species, which can result in adult mortality and also adult changes in behavior, such as nest desertion, and also lowered blood hematocrit and overall reproductive success when it comes to nestlings. And I'm sure there are probably some post-fledging effects that might be occurring as well, though there is little to no data on that. I know that was a lot of introduction. So if you're like, whoa, that's a lot of info, there is so much to learn and study out there when it comes to this old, incredibly interesting relationship between ticks and birds. But if you take nothing away from that, I think it's interesting that when we study these two together, we can have a better understanding of not only bird health, but also human health and also tick movements of well. How are they moving on the landscape? And then also, again, this whole zoonotic cycle of connections between our host community and tick-borne disease risk in humans. And I am interested in particular in exploring the pressures of ticks on birds during their breeding part of the annual cycle. All right, Now, getting into the meat of our questions, I am interested in exploring what is the relationship between tick infestation and nest success? And this is a bit difficult question to answer because there can be many confounding interests and factors along the way. But this is something that I will try to explore as much as I can in this project and still am exploring. So my question is, how does nest height impact tick loads, not just on our adults, but also the nestlings in our target bird species? And I'll do this by identifying tick loads on our nestling adults and differentiating between species and life stage and looking at the abundance of these on our individuals. And I want to determine how tick infestation variance impacts our target species, which include warblers, vireos, and thrushes. Now for our lovely field site. I actually studied birds still here in Ohio, and if you didn't know, part of the Appalachian foothills region actually goes through Ohio, which is interesting. And my site was down in Vinton County at Vinton Furnace State Experimental Forest. It is an area that is dominated with oak and hickory mature trees and was ideal for target nest searching or nest searching of our target species. And before we get into the specific species that we found, when you're thinking about our predictions and our questions, it's important to know what we do know about the movement of ticks on the landscape. So tick questing, which can also be called tick ambushing, which is the movement of ticks within their environment where they put their little front two legs up and wait for a host. And they wait to grab on and then blood feed on that host. And ticks, of course, do have limits to where they can quest. So it differentiates by the life stage of the tick, where that maximum threshold questing height for our adult ticks is up to 1 meter, a half a meter for our nymphs, and then below 30 centimeters for our larvae, which are the youngest life stage. So our question is sort of built around these maximum thresholds for these life stages of ticks, and whether these ticks of their own motility should be able to access the nest or not. And now for our lovely focal species. Our main ground nesters included our ovenbirds and wood-eating warblers, breaking out into our first functional group. And then we had our shrub nesters, which included our hooded warblers. And finally, we had our tree nesters, which included wood thrush and red-eyed vireo. And to break out these functional groups a little bit more, of course, all of our ground nesters occurred at 0 meters. And on the bottom right, you can see one of our incubating female ovenbirds in her namesake oven-shaped Dutch oven nest there on the right. And then the top left, a worm-eating warbler that typically occurs on steeper slopes, simply forming a cup with white pine needles often nestled between kind of overhanging leaf litter, which were our two main ground nesting species. We also had some other incidental ground nesting species as a part of our project, including Kentucky warblers and black and white warblers, which were also a treat. In addition, we have our shrub nesters, which always occurred below 1 meter, between 38 to 88 centimeters at Vinton, and they were typically located in small saplings or in greenbrier shrubbery, and very well hidden as well with their nests of shredded bark and fine grasses. And of course, adding on our tree nesters, typically our red-eyed vireo sample was a little bit higher than our wood thrushes, with our wood thrushes nesting quite close to the trunk, towards the bole of the tree or within the bole of the tree. And then our red-eyed vireos were typically in a hanging cup nest towards the outer foliar edge of the tree. And our wood thrush roam was predominantly in American beech, where red-eyed vireos had a much higher variance in the tree species that they would choose to nest in. And now thinking about these three functional groups in mind, we can think a little bit about our predictions. In this presentation, I mostly focus on the blue box at left, looking at this variance in nest height and how this might impact tick infestation, where I predict that ground nesting and foraging will result in higher tick loads on both our adults and ticks, or not ticks, chicks, and that these nests that have these higher tick loads will have lowered nest survival. And if we look at the box in pink here, you can see if you look at the different species, not all of them forage where they also nest. So this is going to result in some variance that we might see, or that is my prediction, I should say, in that where the bird is foraging also matters how these adults might be picking up ticks and bringing them back to the nest, which is further explained in kind of my hypothesis for why ticks might be present or absent in the nest. First, it could be related to tick behavior itself, where ticks are accessing the nest of their own motility through their chemosensation and then ability to move. And then it could be related to adult bird behavior, where while these birds are foraging, they might be picking up ticks that are also questing, and then the ticks get access to nestlings by dropping off of these adults that are either feeding the nestlings, provisioning the nest, or perhaps incubating. And then finally, I would also like there to be movement potentially for other routes to occur within my thought process, where ticks could be potentially accessing nestlings in some other fashion, like they were present in nesting material or could be dropping off of brood parasitism, which did occur on our sites, or other vertebrates perhaps. Lots of thoughts going around. And now, how did we collect our data? And in my opinion, I love field work so much. So this is one of the most fun parts of bird work for me, and research in general, is how do we operate in the field? Of course, we had to nest search for our species, which I'm sure many of you in AOS know this is not an easy task at all, and it depends on the species. As you can see here on the left, there is a Kentucky warbler at the base of the sapling here, which is one of our incidental species. And sometimes that female won't even leave the nest as you approach her. And these nests can be quite tricky to find. And even our shrub nesting species like the hooded warbler, which were extremely abundant on our plots, can be quite hidden in tangles of greenbrier and young maple and oak saplings. But we did find a variety of nests, 164 of them, and for each nest, we took fate data on these nest cards in order to find our first egg dates and then hatch dates to eventually fate our nests where they succeeded or failed on our landscape. In addition to that, for all of our nests, if they succeeded up into that point, we also checked the nestlings for ticks, as well as took a lot of other measurements as well, including scoring, wing chord, tarsus measurements, and also Coleman measurements as well as mass. So we measured, oh gosh, I think 150 nestlings, or 145 chicks, I should say. And there's some pictures of me actually combing through birds, particularly nestlings, in order to look for ticks. On the nestlings themselves, there was more, though I did check the whole bodies of both adults and nestlings, there was a much greater chance because of the bare areas between emerging feather tracks to potentially find ticks on these chicks. Our adults also got checked as well. And I think it's the pictures themselves don't necessarily do justice of what's occurring, since I think in the photos it looks like they're getting a little haircut. But I have a little video here to demonstrate of what the combing through feathers in order to find ticks looks like. It is actually quite a calm process, and it's amazing that when holding the bird properly with the banjo grip, and calmly with your fine-bladed forceps going through and removing ticks, what a calm process it is. So this is a Kentucky warbler getting tick checked. So in order to check our adult birds and nestling for ticks, we would typically comb through the whole body, going against the grain of the feathers, and also blow on parts of the birds in order to look for where these ticks might be rooted in at the skin. We did not find any ticks that were crawling around on the birds. They were always attached where their hypostome or mouth parts is inserted into the skin. We also did measurements on our adults as well, and which were sometimes helpful for aging and sexing as well. And though our birds in a lot of our images look alarmed, no birds were harmed in the process of this, and we all had our proper permitting and everything we needed. And I had 4 technicians throughout the season to assist me as well, which was wonderful support for this project. And while we were focusing on our main question, we saw some interesting birds along the way too. More variance in breeding birds and body condition that I think I've ever seen at stations I've worked at before. And some interesting observations included this female hooded warbler on the bottom left. I don't know about you, but when I first saw this bird, I would not have guessed this was a female, since females typically have only an olive cap with little to no black, depending on their age. And when I first saw her sitting on the nest, I was like, oh my gosh, are you a woman? But we banded her, and I was amazed to see just how much variance occurs in the plumage and condition of these birds. So I am so grateful for the privilege to get to work with these incredible species. In addition to working with our birds, we also wanted to get an idea of what ticks are expected on the landscape itself so that we could then calculate or look at which species are not only on the landscape, but see if we have any species that are missing on our birds, which might indicate that ticks are having, or ticks do have preferential feeding, but whether it's occurring on our landscape. For example, if we had a lot of a tick species on the landscape and then none on the birds, that would be interesting to know that these ticks potentially are not utilizing these birds as a host. So this was the act of tick sweeping and flagging, where you drag a 1x1 meter cloth behind you that you can see one of my technicians demonstrating here in order to pick up ticks. And you stop the drag occasionally and flip it over, and then check that drag in order to see if you have ticks present on it. And then we collect that in a vial and go and identify it in the lab. We also did a variety of vegetation data taking as well, which I won't go through all of these, but one thing that is important to note is we did choose a lot of these measurements, not only to look at nest site selection and other variables, but also things that might potentially explain tick presence and absence in our nests as well. And in particular, for this presentation today, I will be looking at nest height averaged in our functional groups to see how that might vary with ticks, tick infestation in our nest. Now, for what everybody is interested in, what did we find? And we are still in the process of analyzing a lot of our data. I'm in the midst of spending hours in the lab a day, identifying a lot of ticks to genus and species if possible, as well as constructing my analysis and how I will run things to more adequately explain what we are seeing. But here's what we have so far. For our nest data, we found a total of 164 nests, including 38 ground nesters, 33 shrub nesters, and 93 tree nesting species. We had a variable number of nests in these groups because some species were more likely to be found than others on the landscape based on our skills and also simply the densities of birds and territory sizes that we had on our plots. And now, before we look at actual ticks on our birds, I want to look at the relationship between nest survival and height briefly with you. So what I have for you today are not intense in-depth analyses, but comparisons that are guiding where my analysis is currently heading. So on the left y-axis, we have our percentage daily survival rate of our species. And then we also have on the x-axis, the average nest height split out by species. And if we look here, we have a suggested relationship between daily survival rate and nest height. So for our different functional groups, as that species nest height gets higher, that nest was more likely to survive across species. Our next step for this would certainly be create a more in-depth model, adding species, DSR, and nest height. And now, as we turn to looking at birds on ticks, or excuse me, ticks on birds, that would be a little scary. I'm not quite done with my lab ID, but we certainly have a number of ticks, not too many left to go, so we're getting there. But in terms of the tick species that we found on birds so far, we have found Ixodes scapularis, which is also known as that deer tick that is the vector for Borrelia burgdorferi, the Lyme disease causing agent. and also Haemaphysalis leporus palustris, which is the rabbit tick, which is one of those 12 Ohio species that only feeds on wildlife, which is interesting. And then from our background so far, we have found other Ixodes genus species, and there are other Ixodes that occur besides that black-legged tick. So we still have to see what particular species we have there. And also Amblyomma americanum, also known as the Lone Star tick, that is named for this white spot on the adult. This was a species that we have yet to find on our bird, but makes up more than 90% of the ticks that we were finding on our drags, which is quite interesting. It has been documented in literature that they do not preferentially feed on birds. So this was also an observation that has so far been held up within my study as well. Now, another interesting thing is the tick locations, or the locations of the ticks that we are finding on the birds themselves. On our adults, we always find them around the face. We find them within the ear and around the feathers on the ear, located around or above the eye, along the mustache line, and then along the gape of the birds, including occasionally in the mouth. And then on our ticks on chicks, we find them in a much higher variety of areas, including also that face, and then other places along the body where there are gaps of skin between feather tracts that are emerging or coming in. And now for the ticks on adult birds, to just show you a few more in-depth photos, because who doesn't love pictures of ticks, right? You can see here an example of both larval and nymphal ticks that are located on a wood thrush adult. And that was including one of the ticks that we found inside the mouth, which was quite interesting. And you also find ticks, evidence of ticks feeding on birds besides general attachment, and that these ticks are sometimes in various levels of engorgement on the birds, meaning that they've spent already a certain amount of time on the birds themselves. So when you typically see that kind of grayish color and expanded body frame of the tick in the bottom right-hand corner, is often a tick in some stage of engorgement on that ovenbird. Of course, we also found ticks on our nestlings as well. On this left photo, we have a wood thrush with two of those deer tick nymphs on it here, and each of our ticks was then stored in ethanol to get identified later. So this is quite interesting as well. And as we move back along the body of our chicks, not only did we find them around the face, but as you move back, we also found them along the shoulder, sometimes right at the base of those emerging pins that you can see here in the up-close photos. And typically, in order to find a tick, its head is completely lodged within the bird's skin, and you typically see the kind of flap of the scutum or body of the tick with its legs sticking out, which would be comical if I wasn't a little concerned for the chick. And additionally, as you move back towards the vent, we also found ticks along the back and also on the tibias of our chicks as well. So being held in these photos are two ovenbird chicks with ticks on them. And now that we've looked at some photos of tick infestation, I'd like to talk to you a little bit about our basic comparisons since we're in the middle of our more intensive analysis. So if we look at the percentage of nests with at least one chick infested, so we have our three functional groups and the percentage of nests infested on the left here, you can see our ground species had a higher percentage of nests with at least one chick infested, higher than our tree species, and then also higher than our shrub species. Based on our prediction, I would have expected our tree species to have the lowest percentage of nests with at least one chick infested, simply because they are the highest nests. But that is not something that we saw with our shrub nesters, but did hold true between our ground and tree nesters so far. And then if we look at the percentage of the parents instead of the nest with ticks, we found that our tree nesting parents actually had the highest percentage, so 1 in 5, of likelihood of ticks being on an adult. And if we split that further between the two species that we looked at, I thought it was interesting that there were no ticks at all on our red-eyed vireos, but more than a quarter of our wood thrush adults that we target captured from these nests had ticks on them, which is interesting. And this could have something to do with foraging because red-eyed vireos hardly ever foraging below 1 meter, and where our wood thrushes are predominantly ground foragers, which could be resulting in the wood thrush's foraging, picking up these ticks while they are questing and bringing them back to the nest. Additionally, if we look at tick infestation intensity, or the level infestation, we look at something a little bit different. Level infestation refers to, whoops, I accidentally flipped my screen slightly. The level infestation refers to a quantified measurement of the number of ticks on a host. So if we look at the number of ticks per nestling based on our functional group again, we have on the y-axis the average number of ticks per chick, and then comparing that to our different functional groups. Though we did see our ground nesters were more likely to have ticks prevalent in the nest itself, the wood thrush themselves had more ticks per chick, so a higher infestation intensity, even if ticks were less likely to be in that nest, which is quite interesting. And I think that might have something to do with, or this higher infestation intensity might have something to do with their foraging habits and also larger body size compared to our worm-eating warblers and ovenbirds that make up our ground functional group. But so far, as we proceed with our analysis, this basic comparison does suggest a relationship between tick infestation and nest height. So as our nest height increases, we see our tick infestation decrease. However, in order to further look at this relationship, we will definitely have to do future analysis. So for my next steps in this project, I'm planning on doing an analysis of nest survival versus tick infestation variance, and considering the factor of foraging in this, and doing it at a nest level varying by individual. And of course, I have to finish my identification, that's a hard word, of tick specimens to species of our ticks from our birds, as well as finish identifying all the ticks that we have occurring from our background sweeps as well. But so far, that's the story and that's the direction we're heading. And I still have a few months of analysis to go, but I'm very excited to see where these things take me next. But so far, that's the name of the game, and I'd like to thank you all for listening. I have a lot of people to thank, and they're here on the screen, but I wanted to give a special thanks to the American Ornithological Society for not only giving me 2 travel grants to go to their conferences and also helping fund my research, but being people that I can ask for advice, ask for help, and helping me build connections with other scientists, artists, and people to help promote not only the scientific side of me, but also the artist and personal side. And I'm just very grateful to get to talk here today. Thank you so much. And if you have any questions or comments, I'm so excited to hear them. Thank you.
All right. Thank you very much, Anna. I hope everyone enjoyed that talk as much as I did. Some fantastic visuals, passion, humor. The talk was really great. Uh, and I am looking for any questions that anyone has. You can raise your hand again or type in the chat or the Q&A. So someone who's anonymous is asking, of the ticks found on birds, how many were I. scapularis versus the other species you found?
That's a very good question. I don't have specific numbers for you right now because I still have a few ticks on birds from 2025. But for our Ixodes scapularis, it is mostly Ixodes scapularis, so upwards of above 80%. But these are still, I still have a few ticks left to identify, so stay tuned. But thank you for the question.
Alex Matthews asks, did you notice any differences in infestation by nestling age? Question.
Thank you for the question. We didn't have a whole lot of variance in age simply because we tried to check the nestlings around the same time. before they were too young, and there might not have been much time at all for ticks to potentially um, attach to them. And then, of course, before there could be the chance of force fledging. So we typically checked the nestlings around day 5 or 6, that did change a little bit depending on how long it took them to fledge. But to directly answer your question, that's not something I saw at this time. Though if I had to comment on it, I would say that infestation is more likely the older the nestling is. Simply, it's been potentially exposed. If there are ticks in the nest, it's been um, exposed, or there's been a longer exposure time where attachment could occur. Um, any other questions?
Yeah, Brian Suru asks, since you save the ticks you remove from the birds, are you planning to check the ticks for the prevalence of certain diseases of interest, like Lyme disease? Yeah.
I would love to do that. Originally, when I designed this project, that was something I really wanted to do. And then I realized that I won't be here for 5 years and can't and won't be able to do all that lab work myself. These ticks are going to become a part of resuscitating the tick ecologist lab. So they're going to be identified, properly labeled, and stored. And hopefully there will be enough funding in her lab to check all of them for pathogens. So it's not guaranteed, but it's one of the next steps that we'll get to if we have the time, though it is not something that in my project specifically I will be exploring. But a great question, and certainly something I would love to see happen.
And we have another question. Where do the shrub nesters, which I guess is hooded warbler, in your study forage?
Another great question. Our hooded warblers, in fact, I don't have like exact numbers for foraging, but in my little prediction diagram, they do forage typically at the shrub level as well. So little to no ground foraging occurs in hooded warblers. So they're typically gleaning and encountering invertebrates at the shrub level and not too much higher than that. I did see a few hooded warblers anecdotally that were in the understory, but not frequently.
And I was just wondering, so the wood thrush adults had a lot of ticks, and the red-eyed vireo adults did not have any ticks. Um, but then what about the red-eyed vireo nestlings? They had ticks?
They did not either.
Okay.
Yes. So we had no ticks on our nestling vireos or the adults, which I thought was quite interesting. And actually, one interesting point is that someone else in my lab who also has captured some vireos has also found only one tick on a vireo, and it was a dead tick, which is interesting. So I wonder what's going on there.
Uh, and sort of related to that was because the results were displayed by uh, nest height level in the three categories, was there also a substantial difference between Ovenbird and worm-eating warbler for those? For the lower category, just how there was a substantial difference between wood thrush and red-eyed vireo in the high height category?
Yeah, a good question. In tick infestation, right?
Um,
Or in nest?
yeah.
Okay.
Mhm.
Um, Not that I can recall. I would have to look further. We had a lower sample size of worm-eating warblers, so I
Right.
think we did have a higher, slightly higher infestation rate or of ticks on our nestlings. However, we had a lower sample size, so I don't know if that was really comparable to our oven birds.
Gotcha. Um, Just looking, people have positive comments leaving. Ken Kaufman says, highly valuable, fascinating information, outstanding presentation at every level. Holly Erickson says, thank you so much for the presentation, loved it. So just some positive feedback. And I was
Thank
also
you.
I was also wondering, the range map of the ticks, if I remember correctly, didn't have a distribution in South America. Um, but I think earlier you were talking about birds transporting the ticks over very long distances.
Like this map?
Um, yeah. So w was there evidence of the birds transporting the ticks across the continents? Because I think that that sort of motivated you to target migratory species, right?
Yes. For my project, I focused a little more like the species chosen were what breeders were accessible at my sites, but definitely I'm not familiar with the specific literature between the two continents. I'm not sure if Lyme disease or like that particular bacteria is the best example for intercontinental transfers of disease or ticks. So I'm not the best person to answer that question. But neotropical species or simply species that winter in the southern United States are more of a factor for Lyme disease. So both of those together, rather than only neotropical species bringing things across continents for Lyme disease. Um, but I'm pretty sure I can recall some records in my literature search of mostly exotic ticks being presumably, or let me rephrase that, of ticks being transferred across continents by neotropical birds, but I couldn't name any specific pathogens that that might be occurring with. It's mostly a transference from, for the Lyme disease causing bacteria in particular, from the southern United States to the northern United States and southern
Okay.
Canada, more than from South America to North America. Does that answer your question?
Yes, I think I was just confused about that. But yeah, that totally makes sense, and it makes sense with respect to this map too.
Yeah, I think
Uh,
a better way for me to have stated it rather than simply saying neotropical species were um, species that really have any sort of latitudinal migration that are migrating, even if they don't start in the southern United
I
States,
see.
they're moving farther north. And of course, those dynamics change with different tick species, different diseases. I only mentioned one of, gosh, half a dozen other relevant pathogens that this is occurring to just in our region, much less globally. There's a lot that I am not an expert on with this.
Yeah, that's great. And they could transport them not necessarily from their wintering ground, but as they're returning, as they're coming up, right?
Yeah, they
So
could come up at stopover sites as well.
passing through. Um, yeah. So I just want to make sure I got everyone. Okay. Uh, were the significant differences in percentages statistically significant? I think this is something that you're probably working on as you iron out the analyses.
Yes,
Okay.
I will have to admit that I have a lot of comparisons right now where I have not done a lot of the tests that I would like to do at this point, and I am like in the middle of, and it was too difficult for me to pull myself out of the weeds and add a lot of that and be able to adequately um, interpret it for the audience today, even though I'm in the middle of it right now. So I am sticking with my comparisons that are already a couple, I wouldn't say weeks old, but because I am in the middle of it, I don't want to, um, what's the word? Create an interpretation right now that is potentially inaccurate. So I would like to say that these comparisons I have right now have not been through the statistical test yet. So you are correct in asking that, and I would like to know that as well. And I'm currently working on that.
And I got one more question for you, Anna. What was it? Oh yeah. So if height could be a factor in in how much tick infestation there is, um, can you do that? You did that between species using these three categories, but is there more tick infestation within a species with depending on how high their nest is? Especially, was it the wood thrush that had the most variable height for its
Mm-hmm.
nest?
Yeah. Yes. We originally looked at this with hooded warblers because because they're kind of between this 30 centimeters to 80 centimeters, we were wondering if some live stages might drop out as the nest got higher. And we didn't see any change interspecies with the hooded warbler. And that's something that I actually haven't looked at in the wood thrush yet. So that is a good suggestion. And I will certainly investigate that further, but not with the hooded warbler so far.
Got you. All right. Well, I think that's it. That's a lot of questions. And really great presentation again. Um, and we want to thank everybody for being here. Again, we'll have next week off actually, so we will rejoin in 2 weeks and look forward, as Kamal Islam says here, we're going to look forward to reading, Anna, your published findings when you've
Yes.
completed your research. So thanks again, and we will see you next time. Bye,
Yeah.
guys.
I appreciate the questions. You're always welcome to send me an email if you have any suggestions, since I am still in the process of investigating my findings.
And
Thank
Anna's contact is right there on the
you.
slide. So, all right, guys.